Now, we've learned a little bit about DNA. So, DNA is the code. DNA has no chemical properties like carrying out reactions. It's just copied by RNA into RNA, and then the RNA is then translated into chemistry in terms of proteins. Okay, but, and I've mentioned this before, instead of a gene being expressed on or off, it's not. It's like a dimmer switch. Okay, more gene, more DNA product, or more protein product, less protein product. You can have the same gene on in different cells, but the amount of protein product is going to be different because the proteins are doing different things. It's the same protein. Now, you can also have the same gene, and it actually can produce a bunch of different related proteins. So it was originally thought, before we had the technology to read DNA and transcribe it into and translate it into protein, that, oh, there's a gene for each kind of protein. Well, there's a couple of 100,000, probably more proteins, and by the way, once you get a protein, they can be modified after that. So it has chemical modifications and different chemical properties. And you can also, it turns out, get different forms of the protein by transcribing parts of the gene and skipping others. This was found out because it turns out human beings only have about 20,000 protein coding genes. 20,000. There are hundreds of thousands of different proteins. So suddenly people started, a light came on, obviously, that you mean you can get different proteins from the same gene? Yes, you can. You can end up with a whole bunch of different versions of the same protein that have different properties. What is it called? It'll come to me. Anyway, we know what an allele is. This is a review. Oh, we've already done this. We already did. Okay, here we go. So, what kind of cell is this? Is this a eukaryote or a prokaryote, or impossible to tell? Eukaryote. Okay. Why? Well, it's got a nucleus. It's got a mitochondrion and a bunch of other stuff. So, in this case, with the eukaryote, the transcription and translation has been separated. The transcription happens here. The translation happens out in the rest of the cell. Bacteria aren't like that. Transcription and translation all happen in the same place. Okay, so what's the complementary sequence to this? Let's start with okay. What's that? G. Okay, good. Okay, so now we've already talked a little bit about this. Double-stranded DNA molecule. We have DNA replication. What's the name of the enzyme? DNA polymerase. Okay. And what it does is, when it unzips, is it takes the corresponding nucleotide from the other side. So, an investigator finds a small amount of tissue at a crime scene. What procedure will a forensic scientist perform to amplify the small amount of DNA? PCR. PCR. Okay. This is all just memorization. Okay. Okay. Why are STRs so useful in forensics? Because you can tie in relationships between people, living people or dead people and living people, and figure out who's what. Okay. Gel electrophoresis. We already did this. So here's our DNAs, different fragment sizes, and we have, let's say, a whole bunch of different individuals. We put them in the wells, apply an electrical current. Short fragments go farther. Large fragments go shorter. Okay? Okay. A chromosome is a long DNA molecule. In fact, in terms of all our chromosomes, there's like 3 billion base pairs. Not all of those base pairs code for a specific protein. So a gene is actually a part that actually codes for a specific protein. In other words, we can transcribe it into RNA and then translate it into a protein. It turns out that you can do a lot of other things with DNA. It also codes for a whole bunch of different species of RNAs that have nothing to do with transcription and translation. These different species of RNAs actually are used to regulate gene expression, like turning up the dial, turning down the dial. Okay? In fact, they're actually used to stop gene expression. So what they do is these single strands of RNA glom onto a strand of transcription DNA or transcription RNA and then stop it from being transcribed and translated. There's a whole bunch of mechanisms that go into what's called gene regulation, and it regulates gene expression. And so our DNA not only codes for DNA, it does a lot of other stuff. I mean, codes for proteins, but it does some other stuff too. Because, frankly, a lot of the structural genes that we have are highly concerned across millions, all the different species. Okay, we use many of the same genes that have been perhaps a little modified that a fruit fly uses. But the difference is in how they're transcribed and translated, the amount that's translated into protein, when certain things happen. So genes themselves really don't do anything. They don't have any, they don't actually change any chemical reactions. RNA, they found some organisms that are RNA organisms that do have that kind of a function, but for the most part. Okay, so these are different amino acids, and the chromosome, of course, we've already gone through the going. Okay, now, synthesis of mRNA is called messenger RNA. This is what goes on inside the nucleus. Oops. Okay? And then this is the cytoplasm. So mRNA is a transcript from DNA. It's a single strand of RNA that corresponds to the DNA, to the complement of one strand of the DNA. The mRNA, messenger RNA, is then transported from here out into the cytoplasm. And then it is fed into the ribosome and read, and these are the amino acids. They're actually floating around. So some of these amino acids we make ourselves. Others we can only get from our food. So remember, what is it called that amino acids that we get from our food? Good. I forgot myself, so I'm happy for you. Okay. Now, then it turns out the ribosome takes these amino acids and strings them together according to what is read along the mRNA. Okay? And that's the protein synthesis that's actually going on. Okay. So remember, this is the order in which things go. DNA, transcription, it's transcribed from DNA into messenger RNA, and then that's ferried out of the nucleus and into the cytoplasm, and a ribosome then translates that strand of RNA into a protein by stringing together amino acids in a specific order. Got that? You sure? Okay. You can always go back and revisit this, but I want you to make sure. We'll have some questions about, oh, which order does it go in? From, is it mRNA to then DNA to then to a protein, or is it, right? So you want to make sure you have that down. Okay. So a transcription and then translation. Transcribe is to write, type, and print. Okay? And then transfer information from one place to another. And then translation to turn from, let's say, one language into another. I guess it's not really a good heuristic model for thinking about it. Just remember, with mRNA, you're getting a copy of the DNA, except it's in an RNA form that can then be translated into a protein. So, in terms of gene expression, how is it that if the whole genome is in each cell and in the nucleus of each of our cells, that means all 20,000 genes, all the different base pairs, exactly the same unless there's been a few mutations. How is it that we get muscle cells and only certain genes are actually translated in muscle cells and others aren't? Well, it's all about gene expression, about how much protein product is made in different cells. There's going to be certain genes that are always going to be translated and transcribed in all cells because they have certain things that they have to do. Part of it is they have to be transcribed and translated, which takes proteins to do some of that stuff, like DNA polymerase. That's an enzyme, it's a protein. But each of these cells has different functions. A muscle cell has much different functions than a nerve cell. They both have a nucleus, can't really see it in a nerve cell, but they look different They're made up of different versions of proteins and amounts of proteins, and they work differently, they function differently. So, all of your cells have the same exact number of instructions. In other words, each cell doesn't just contain the genes that are coded for that cell's proteins. Each cell contains the whole genome. It's, I guess you'd call it a manual, but it's not really quite like that. Because transcription and translation, in other words, gene regulation, it's not just, there's an interplay between what's going on in biochemistry and then what's going on in terms of the environment. Different things are expressed in different cells at different times. If a monster comes in here, there is going to be certain cells, or there's going to be, first of all, there'll be some signals in your brain that go, ah! It'll ramp up adrenaline, which is a hormone, so it'll turn on all these genes that get these cells to produce the hormone adrenaline, and a bunch of other things are going to happen. That's gene regulation, happening all the time. Different things are happening. When you go to sleep, the amount of protein product in different cells changes. Now, you need, well, for diploid organisms like ourselves, where we have a copy from mom and pop, and there's some crossing over in terms of mixing up genes, we need an egg and a sperm. Bacteria don't use this. They just make direct copies of themselves. In other words, they make clones of themselves. They just replicate each other, themselves, and all of a sudden there's 2 bacteria, 4, 8, 16 exponential growth. It turns out that bacteria can also exchange genetic material between them. It's called conjugation. And in fact, bacteria that are different, we'll say species, often share DNA with each other. That seems kind of strange, but that's one of the reasons that antibiotic resistance has become so widespread, because not only are there mutations that give a bacteria, this is off topic a little bit, but a bacteria a little bit of resistance, but that bacteria can then share it with another bacteria. But back to diploids. So each egg is haploid, and each sperm is haploid. That means there's only one, remember we talked about homologous chromosome pairs? There's 23 different chromosomes, and each of us have a pair of each. That means chromosome 1, we have a pair from mom and a pair from pop. When those two come together and there's fertilization, we've restored diploidy. We've got a zygote that now has 23 pairs instead of 23 individual chromosomes. Okay. Cell type, we'll say muscle cell. Transcription of RNA, translation into protein A. We've got gene A, it's being expressed. Something has turned it on to start producing protein product. The DNA is transcribed into RNA. This is taken out of the nucleus and into the cytoplasm, where then it's translated into a protein by stringing together all these different amino acids. So when you have 3 nucleotides of DNA that codes for a specific amino acid. And then proteins are made up of amino acids. Just so you know, you can have a protein with the same amino acids, but it's 3D. How it's actually folded up can be different, and it can have a completely different function. Okay, so this is still muscle cell. We have transcription, not very much. So in one we have a lot of protein A, and here we just have a very little bit of protein B, being from transcription to translation. This is still a muscle cell, and we've got kind of a moderate amount between the two, okay? And then there's nothing being transcribed and translated into protein D, okay? So we can actually not only read the genetic code DNA, we can also read the transcriptome, which is going the amount of RNA DNA has been transcribed into, to see how much gene expression is actually going on. And we can also look at what's called the proteome and see, you can tie it all together. Now, here's a neuron. Gene A and protein A looks like the same thing. Okay. B is off. There's not quite as much of C as there is up in the muscle cell. And then D is also transcribed and translated. So that's how cells and cell types and different tissues are differentiated. The structural components of each of these proteins, they might be slightly different, as I mentioned, because of we can have a gene coding for more than one related protein, but the amount of protein is regulated in terms of how much gene product is actually made. So there's some gene products that are only made early in development. So we have a zygote, it's a single cell. Eventually it's going to divide and differentiate into a human being, an embryo. It's going to be born, it's still developing. There's going to be a lot of genes that are being used that will not be used after that. If, in fact, those genes are turned on and not turned off like they should be, then you're going to probably have a mutation that's going to result in something bad happening. So, instead of an on-off switch, it's more like that. The part of the gene that actually codes for the amino acids is called the coding region. The promoter region is the part that regulates how much coding is going on. As it turns out, it's not always upstream. You can actually have promoter regions or gene regulatory regions on different chromosomes for genes on another chromosome. So when we saw those pictures of the different chromosomes, well, when they're not being copied, they're actually all stretched out, and all kinds of things are happening in terms of biochemical reactions. Here's a single strand of DNA, a double strand, okay. And we'll say that right here, there's, let's say, 1,000 different base pairs. This is the actual gene that's going to be translated. Up above this, if we're going from first to last, there's going to be a promoter region. And things are going to, there's actually going to be proteins that bind to the promoter region that then have downstream effects on how much of a protein product is actually made. Okay? So let's go back. So here we are back with lactase. So we have lactose, that's for milk. It's a disaccharide. It's made up of lactose and galactose. We have an enzyme. So it's a protein. It's got certain regions where reactions actually happen. So this is the coding region. Now, the thing is, there's something called RNA polymerase that binds to the promoter region of a gene and then synthesizes a transcript of that gene. So here we have this promoter. So right here is the DNA being transcribed into RNA. Here's a promoter region. So this is, I guess, what's called the central dogma. This is that DNA is always transcribed into mRNA, which is then translated. You can't go backwards. You can't go from a protein, well, you can in your mind, but in terms of what actually happens in nature, you can't take a protein and then turn it back into DNA. We don't have the machinery for actually doing that. But the promoter regions, or any region that regulates gene expression, that's where really everything is happening. That's where disease is happening a lot of times. In regions where transcription and translation are kind of messed up. So you can have a good structural protein and then it's not being regulated properly. Okay, a little bit more on translation. Here we have, okay, things are going this way. This is our mRNA. Okay, I'll have to remember to go back and put that in. Okay, so transfer RNA. This is the translation part. The DNA has already been transcribed into the RNA. That is in the, this is in the ribosome right now, right? This is the anticodon, which will then eventually has a region of internal complementarity that will bind only to a specific amino acid. A codon is always 3 letters, 3 nucleotides. A, C, G, and U if it's mRNA, or T if it's DNA. So transfer RNAs are the interpreters of the actual genetic code. It translates the genetic code into an actual protein. Okay, this is, let's see, this is, I'll just give you an idea here. Okay, I want to show you something. Okay, this is the first base. We're looking at RNA here. you can either, the first base is this strand right there. The second base is that strand. And the third base is that. Okay. When there's a change in a first base, you are generally going to have a change in the amino acid. So, in terms of U, you can have a first base that will, depending on what's in the second, you can either have, or the third, you can have phenylalanine or leucine. You can also here, with a C, also get leucine. Then you can also get a different amino acid, isoleucine, methionine, that's the start codon, and valine, another amino acid. When there's a change in the second base, that means the middle one. If it's a C, you get UC, and then some third base, you get serine, proline, threonine, alanine. Okay? And then here, of course, instead of a C, there's an A. And then finally, a third base. A third base is actually redundant. A lot of times you can get the same amino acid with the same third base. So, cysteine, let's see, glycine. So you can get what's called fourth time redundant. Well, anyway, I'll get to it. So there's a redundant part of the codon, which is the third codon, which can oftentimes be a different codon, but you'll still get the same RNA, amino acid. But then changes in the first and second base tend to give you different amino acids.